So, I like to kind of look up the guests and like see kind of what they did before, kind of like what their education was and that kind of stuff. And whilst doing that, I found an interesting website. Are you familiar with the website Stangl.Stangl.eu?
No, it sounds funny. No, I'm not.
It's a website that I'm assuming someone with the surname Stangl made about all the famous people called Stangl.
Oh, I see.
It's a really long list of people who are called Stangl with surname.
Well, yeah, let's hope I'm gonna end up there sometime.
You are. That's how I found the website. You're on it. Yeah.
No, I have to. Now I have to write that down. I have to Google that definitely. Funny.
Yeah, it says Matthias Stangl, postdoc Forscher. So it's a German website, and it briefly mentions in 2 sentences kind of what you're doing.
Exciting. Well, now I'm curious.
So you're not familiar with that website?
No, I'm not. And I didn't create it. So no.
Yeah, I think a guy called Werner Stangl might have created it. I'm not sure. Yeah. Anyway, so you're not related to.
No, I'm not. No. I'm definitely gonna look it up. But no.
Is it a super common name? I mean, I don't know anyone apart. It sounds like a kind of Bavarian Austrian name.
It is. I am Austrian actually. So and it is common in Austria, but as soon as you leave Austria, Bavaria, it's kind of not very common. And but yeah, so where I grew up, it's a couple of people. Yeah, it is kind of a common name. In Styria, that's like in the southeast of Austria. Mm-hmm. A lot of like a mountain area, and it's
so like really, stereotypical Austria.
Yes, very, very much so.
Okay,
yeah.
Cool. Yeah, by the way, there's lots of great people called Stangl, lots of chess champions, Kegelmeister.
Oh yeah, that sounds very Austrian too. Yeah, I see.
Skatmeisterin. Anyway, so let's actually talk some science and some research then. So you have the 2 main papers that or the 2 papers of yours on this topic that I've read are the current biology one and the Nature Communications one. I think it makes sense to start with the current biology one. It seems to me that, it's both the historical and also conceptual kind of sensible path. Can you maybe like in, I don't know, a minute or 2, just briefly kind of explain what you did in that study, maybe what the participants did and kind of what you found?
Yeah, sure. So the main idea, what we wanted to look at is if the grid cell system, or, as far as we can measure that in humans, we're talking about grid cell-like representations because we don't have access to single neurons, or we sometimes have, but not typically. With like fMRI, we can only measure population activity. So we wanted to see if the grid cell system was impaired in older adults. And this was especially because, so the main reason why we wanted to do this is we wanted to see why navigation abilities are impaired in older age. We wanted to find potential causes of these impairments. And if you want to do that, if you want to research that, then there really is no way around grid cells, because the grid cells are just such a key player in the navigation system. So, and also grid cells are mainly located in the entorhinal cortex, which is one of the very first regions where neurodegenerative processes start, like tau pathology and other things. They start in the entorhinal cortex. So we just felt it makes a lot of sense to look at whether impaired grid cell activation could be one of the key reasons for navigational decline in old age. To test this very, very simple idea, actually, we used fMRI, as I said before. So we asked people to, perform a navigation task in the scanner. This was all virtual reality. So they moved around using like a joystick while they were in the scanner. They moved around in a virtual environment, and the task was also very simple. They just had to learn locations within this virtual room they were in. And after a while, when they had learned, like whatever, the soccer ball was in this corner of the room or whatever, then they were just repeatedly asked again and again to navigate to this place where they had learned that the soccer ball was. And they did this for like a long time. So it was probably not the most exciting task, but it allowed us to get a lot of data to then look at what these grid cells do while our participants performed the task.
So maybe actually one thing just before I forget it. So I did do an interview with Nicola Axmacher. It's, I don't know, episode 38 or something like that. So just if this whole topic is interesting to the listeners, there's an entire episode on their science paper about doing this with basically grid cells and path integration and that kind of stuff, and spatial navigation with people who are at a genetic risk of developing Alzheimer's. So just to mention that briefly, if this is interesting, there's another entire episode on this. But yeah, so it seems to me there's kind of like three main findings, right, in some sense. So the first is the grid cell differences, I guess you could say reduction in older people compared to younger people. The second is the path integration differences, right? And the third is kind of the combination of the two.
Yes, that's correct. I mean, we did know before, or it has been shown several times that path integration is impaired in older adults. So that wasn't really like a new finding, but we were really interested first to see whether the grid cell system might be, impaired or grid cell-like representations might be reduced or compromised in older age. And then we wanted, kind of as a next step, we wanted to see whether this could actually cause navigational impairments. And so we looked at path integration in particular. It wasn't surprising that older adults were worse in path integration than our younger adults, but our hypothesis really were confirmed when we saw that those older adults who were worse in path integration were also the ones who were showing the reduced grid cell-like representations. Now, obviously, this is not like a proof for like a causal relationship, but it just shows us that these compromised grid cell-like representations could actually really serve as an explanation for navigational deficits or path integration impairments in old age.
Mm-hmm. I mean, so maybe to start or talk about the grid cell aspect here, the thing that was kind of most interesting to me, I think, it was this parallel finding to the Kunz 2015 science paper, where you found that the grid cell activity was, if I understand it or remember correctly, that the main reason for the reduction in grid cell activity was because it just wasn't as stable over time in terms of the direction in which the grid cells or the grid cell-like representation fired between the first and the second half of the study. So kind of what do you make of that? Because I have to admit, I don't know much about this temporal stability of grid cells. Can you maybe talk a bit more about kind of, what we know as a field about this?
So I would say about the temporal stability, we don't know too much, but it is very obvious that a pattern forms. So a couple of findings come together, and Lukas Kunz, Nicola Axmacher and their colleagues paper that was like a first step, really showing that temporal stability seems to be something that is impaired, meaning that what we can measure is the orientation of this firing map. So basically the orientation of our internal coordinate system that grid cells form, that this is it's unstable. It kind of shifts over time. And so in rodent studies, it has been shown typically that, this is kind of stable. Like when you would go into the same environment multiple times, it seems that this kind of coordinate system that grid cells form, that it seems to be pretty much anchored to something. We don't know exactly what it is, but it seems to be pretty stable. But what we see is that, and that's the same in our paper and in the paper led by Lukas Kunz, is that when these coordinate systems are less stable, when the temporal stability kind of is reduced, then this is kind of an indicator for reduced performance. And they see this already in younger adults who are at risk for developing Alzheimer's. And we see really the same thing. So it's very strikingly similar that we see the same thing in older adults, and particularly in those older adults who are worse in actual navigation performance or in path integration.
Yeah, I mean, so I kind of also got the idea from your reading paper that this is something that's pretty new. There's just not that much done on it. I mean, so I guess maybe I'm asking you to speculate a bit here. And I guess to some extent you do that in the paper by suggesting that head direction cells maybe have something to do with it. Also, like one thing that's not entirely clear to me is why would it be a problem if grid cells aren't super stable over time as long as they can do it in a given moment? If I know where I'm right now, why is it a problem that 5 minutes later it's different grid cells doing it? Yeah, I was just surprised, I guess, by the finding.
Oh, I see. So I wouldn't interpret this as, like just a different grid cell takes over with a different orientation. How I would interpret this is that the firing of a single cell, but this is, as you said, this is speculation, right? We don't know that. We don't have access to this kind of information in fMRI. But how I would interpret this is that a single cell, that the firing of a single cell is just not stable. These cells fire at particular locations in the environment. They form this kind of pattern. So if you look at the fMRI data that we see, I would speculate that the single cell firing, that is the underlying basis for what we see in fMRI, that the single cell firing is just not stable, meaning that this one particular grid cell fires at a couple of locations in the room, but then like 10 minutes later, it's firing at slightly different, slightly offset locations, which it's really not clear why this is causing an issue, but we see that this it's not what kind of a normal, very prototypical grid cell would do, or at least what we expect it to do. And figuring out why this is the problem, and then as a next step, what can we do about it, is kind of really the next steps. It's kind of the $1 million questions that we don't know.
Yeah, it seems like you and the other group or groups on that paper added like a whole new question for people doing animal studies, particularly like animal models of neurodegenerative diseases, to kind of see like, yeah, whether it's the case there too, and why, and yeah, all that kind of stuff.
I will say that I think it might. So there are some papers suggesting that it's more like a general mechanism related to age-related decline, at least in spatially tuned cell firing. I can't remember exactly what papers these were, but I know I have seen papers also showing similar effects in place cells, for example. And I'm not sure about head direction cells, but definitely in place cells, there was also, I think it's Carol Barnes, who had a couple of studies on that, showing that also place cells have this kind of, these place cells seem to do their job. They fire at a particular location in the room. It's just not stable over time. And in younger or in healthy or in high performing individuals, you see that these firing patterns are just more stable. So it might be that this is not something typical to grid cells. It could be that it's just a general age-related issue in the firing of, neurons, or at least in the firing of spatially tuned neurons, that they actually function, but they are not functioning in a stable way over time.
Right. Although I guess isn't the idea that place cells get their information from grid cells? So kind of if one isn't as stable, then it kind of makes sense that the other, as a downstream effect, wouldn't be as stable either, right?
It's true. It's possible. I have to say that it's also another open question. So initially, many people thought that this is the case, that place cells are formed by input from the entorhinal cortex, particularly grid cells. But also turned out over the last couple of years that it's probably more complicated than that. Yes, I agree. But you could say the same thing also about grid cells, and we're trying to make this point also in the paper. We don't know whether grid cells are kind of the source of the issue, right? It could be that the information that is delivered to grid cells or to the grid cell system, to the entorhinal cortex, it could be that the information is already kind of compromised before that stage. It could be that the information that is delivered to the grid cell system is already, not stable, and that's why grid cells maybe can't even form this stable pattern, because, potentially the head direction input that is then delivered to the grid cell system, which obviously is important if you think about your anchoring in an environment. You probably, the head direction system plays an important role. It could be that this is where it starts, or even in the sensory processing. So it's hard to say where it starts, but definitely we see in grid cells, or in grid cell-like representations, we see that at this stage, the information and the information processing is already compromised.
Yeah, it's funny. I guess you have this like multiple streams of different things influencing each other, and you've kind of found in the middle this one thing that doesn't work, and now it's like no idea whether it's the thing or anything that happened before, or whatever.
Exactly. This to be honest, this was one of the reasons also why we want to look into exactly this question also more with like the computational modeling study. We haven't talked about this yet, but this was one of the key reasons we at this point knew, okay, there is something going wrong in the grid cell system. But there are just so many other variables that we haven't, like even considered, or it's just a complex picture. So we wanted to also take other variables into account. And so exactly what you said, it's just a complicated picture. Seeing that the issue is already present at the grid cell system doesn't mean that it's the source.
Yeah, definitely. Maybe shall we just go there then and talk about the Nature Communications paper?
Yes, of course.
I have to admit, this is the one I'm probably least familiar with of the three we're going to discuss today. I guess in part because the details here are in the modeling, which is probably the point, my weaker point of these studies. Yeah, I guess we can still kind of discuss it and see kind of where we go. So maybe again, do you want to kind of briefly set the stage of what this paper did, or maybe how you got? I guess you already did, but kind of what were the main findings?
Of course, yeah. I mean, the main question was very similar to the paper that we talked about already. It's like, why is navigation impaired in old age? So we do see this many, many times, but still we don't know what kind of processes, what kind of neural mechanisms, or what variables do actually play a role in this age-related navigational decline. And again, what I said initially also is navigation is just such a complex function, such a complex ability. So we also just looked at one specific subcomponent, which is path integration here, which is just an important function in navigation, but it's just one element where you keep track of where you are by integrating self-motion cues. And so we already knew from this current biology paper that we've discussed, we already knew that the grid cell system does play an important role, and this has also been hypothesized and shown many times before. But also we knew, or at least we thought, there are just many, many other factors and variables that could play a role, which were not, like, we didn't look at these yet. So now, having closely looked at the grid cell system also, we wanted to know what about the other variables. For example, in navigation and also in path integration, for example, memory also plays an important role because you need to, continuously maintain and update a location estimate, information about where you are. And for that, you also need to remember where you've been, 3 seconds ago. These kind of things. Also, there are just many, many other variables involved, like you could over or underestimate your self-motion speed. And this is why your location estimate might be imprecise, for example. Or something very, very different, like what we call reporting noise, for example. So it could be that people actually, I'm not sure how plausible this is, but theoretically it could be that someone is really, really good at path integration, really good at keeping track of where they are. It's just when you ask them about it, their report could be kind of noisy, could be kind of wrong, right? Which doesn't have anything to do with path integration per se. It's just they're not able to translate this internal estimate to kind of an output. Or there could be something which we were obviously most interested in. It's kind of, we call it internal noise. So this is a vague term, but this is what we mean by the actual path integration computations, which is where you take sensory information and you compute your location estimate based on the change in sensory information. So you basically move, you see that you move, you feel that you move, you take that information and then you compute your new position. And this process could also be noisy or could be imperfect. So, and we wanted to know, so now knowing that older adults are impaired in path integration, we didn't only want to know like how strong is this impairment. We wanted to know what processes are going wrong, what could be the sources and the causes for path integration decline in older age. And so what we did is we teamed up with computational neuroscientists, really great scientists who built this computational model. So this was Ila Fiete and Ingmar Kanitscheider. They were at the University of Texas in Austin, and Ila Fiete is now at MIT. And so we worked with them, and they built this computational model, which takes the variables that I've explained and a couple of others into account and assumes that this location estimate that you continuously update and maintain during movement, and that this location estimate could be corrupted by these sources of error, like what I said, a memory component, this internal noise, this reporting errors, these kind of things. And so this model allowed us to estimate for each individual participant, to estimate what was the contribution of each error source on their total path integration errors. So we disentangled these different influences of the different possible error sources, and then wanted to look at which sources are actually driving the navigational errors, which are the major causes of path integration errors in both in younger adults and in older adults. The first thing that we found was very surprising, because previously it always so a lot of studies have shown already that path integration is impaired in older adults, as I said. And always they were speculating or assuming that memory plays an important role, right? That probably like memory is impaired in older age, and this is obviously an important component for path integration. So the assumption was that this memory component, which is also called memory decay or memory leak, kind of a forgetting of where you've been a little while ago, that this drives these navigational errors. And what we saw in our task and in our model is that this memory component actually did not play an important role at all. It was I think even like one of the weakest sources of error in our participants, and this is true for both young and older adults. So this was a major finding, even though it was a non-finding really, but it was interesting that it was so different to what everybody had assumed and expected.
I mean, I guess the assumption is always that memory gets worse with age, right? I mean, that's
exactly
the kind of obvious thing, but.
Absolutely. And this I think is why everybody was under the belief that memory would play an important role. But for this particular aspect of navigation, it seemed that memory is not the driving factor, and there is something else. And we also saw that it's not these biases and like reporting errors or over underestimation of speed or something like that. We saw that it's really this internal noise that is the main source of error, both in young and in older adults, which is in a way also in line with what we said earlier about, the grid cell system and these neural processes. So it really shows that the actual path integration computations are impaired. In this study, we didn't use fMRI or any other neural recording, so we can't really say whether it's originating in the grid cell system or where it really originates, but we can tell it's really the path integration computation that goes wrong. It's like at some stage, starting from where you get the sensory information, where you process the sensory information, and then it reaches probably like the grid cell or the path integration system. Somewhere on that stage or on these stages, this seems to be like the main source of error, and not so much the other variables that we looked into.
And I guess the thing that, I mean, you kind of alluded to this already, but the thing that I found interesting is that it seems to me that correct me if I'm wrong here, but like a very, very brief summary would be basically that older and younger people make the same kind of errors, but older people make more of them. Is that fair to say? That like basically, I think you said they both kind of have the same kind of reason for error, just that older people do a bit more of it, which I don't know. Initially, I also thought, like, in terms of clinical application, this probably is not good. Like, you'd hope for a specific difference rather than just, the same thing.
I see. Yeah, no, but I agree. I like that summary a lot. It's actually a really good description of what we think is happening. It's not like qualitatively very different between younger and older adults. Yes, they make the same kinds of errors, and in both like age groups, a lot of variables like memory are not driving the navigational errors, but older adults make more of this. So it seems like the problems, I mean, this is all like not a pathological level, but kind of the problems or the sources of error that are already present at younger age just get worse with older age. And, it could be that this is happening in the grid cell system. But again, as I said before, it could also be that it happens before. It could be like the sensory input to the grid cell system. It could be head direction information, all these kind of things. But yeah, it seems to be the major source of error that is already present in younger adults seems to get worse with age.
Yeah, I mean, maybe whilst you were, so one main point kind of about this one I had, or not necessarily main point, but one thing I wanted to ask about that you kind of alluded to just now is, this kind of like, where exactly does this noise or error come from in that sense? And one thing I just from like reading the abstract, I think, or something, my initial thought was like, okay, like how does this relate to proprioception or something like that? Because in some sense, proprioception being kind of your awareness of where your body is in space, I guess. I hope that's a good description. And so then I kind of, whilst I was thinking about this, okay, like, is there anything about grid cells and proprioception? Or, because as far as I'm aware, these are they're different research areas, but there's a lot of overlap between people who do proprioception and bodily awareness and spatial navigation, right? It's not that different, but I'm unaware of any studies on this. I'm not in the field, but do you know of anything?
So, the question you're asking is a really good one, is a really important one. It's like, where does this noise come from, right? And unfortunately, it's a beautiful question, but I don't have a good answer to it. Particularly like with our study. Yeah. We didn't have any neural recordings even, right? So this is the information that we get from a computational model, just looking at behavior. So what we can say is we can exclude a couple of variables in this study, in terms of them not being like super important. And we can really say, okay, it's the path integration computations that drive these errors. But at what stage of these processes, at what stage exactly this noise comes in, we don't know. And it's very possible that what you said, that I'm not sure it's proprioception per se. I think there are a couple of other good candidates, but yes, I 100% agree that the sensory information that you get, the processing of the sensory information even before it reaches the path integrator, that this can, and I think it's likely that this plays an important role as well. And I'm not 100% sure, but I think there are studies pointing towards like vestibular information, at least in rodents, vestibular information playing an important role. I think there are studies showing that the grid cell pattern breaks down when you manipulate vestibular information, or when vestibular information is reduced to the animals. So at the same time, I will say that reading these papers, it's obviously very interesting, but it's also not clear how it translates to humans, because when it comes to exactly this, like processing of sensory information about where we are, I'm under the belief that this is something very different between humans and rodents, for example, right? So in humans, we I think use different sensory cues, different sensory information to get an understanding of where we are. We are probably also easier able to imagine being somewhere where we are not, or we don't have to go to a specific place to, like explore it. We can just look at it. So I think humans are much more visually driven.
I was about to ask, do you mean like humans more use vision more and other animals use, especially smell a lot more and that kind of stuff? Or?
Exactly. So I think the kind of sensory information that we use to keep track of where we are is probably different in different species and in humans in particular. So I think what we know from rodent studies might not be like exactly the same when we look at humans. And also I think this is an important point also, because if these processes, like also the grid cell system and the processing of sensory information, would be the same in humans as it was in rodents, we would not actually even expect to see any grid cell-like representations in fMRI, for example, because these participants are not moving at all, right? They are in a scanner, or with electrophysiological methods. We see that pattern also when people are like in their hospital beds, not moving at all. They just navigate pretty much in a computer game with a joystick. So you don't have that sensory information. The only sensory information you have is pretty much vision, but we still see, grid cell, firing patterns. We see grid cell-like representations. So that already tells us that these systems might use different information in humans than they do in rodents, which makes it even more difficult to speculate about what is the driving source in humans now. But again, it's perfectly possible that it is sensory information. It is proprioception or vestibular cues would be definitely something to look at, but also on other sources of information.
Okay, I know you're not a rodent researcher, but this is just a question that occurred to me, so maybe you have an answer, maybe not. It just occurred to me that, I mean, I'm assuming that rodents move a lot around in the dark, if you're in a cave or whatever, or at night and that kind of stuff. But as far as I'm aware, I think most studies are done in light, right? I don't know, it just occurred to me like whether we're in some sense maybe, I guess one question is whether there's a difference in navigational strategies for rodents, for example, or whether it doesn't make a difference. They just use a different sensory modality. When it's dark, they maybe use their sense of smell more than their sense of vision. But it also just occurred to me, like, are we, in some sense, maybe even testing rats in an environment they're not actually that familiar with? I mean, I don't know what the etiology exactly of rats is and how much they actually spend in light and dark. Do you know anything about this? I don't know. It just occurred to me whilst you said that.
Yeah, no, it's a good point. And I think that's also part of the reason why it is so important to take the findings that we see in rodents, but then, or in other animals, and then try to translate it to humans, but also kind of try to be a little bit unbiased, right? Because things are different. We don't know exactly, like what you said, I don't have the answer to that because as you said, I'm not a rodent researcher, so I don't know how natural it is for a rodent to run around in light and dark in different conditions or in these cages. But whatever we find, we don't know exactly whether it's going to be the same in humans, and so that's something to look at very carefully. I do know that grid cells have been shown in rodents also to show their typical firing also in darkness. So it has been shown in darkness and in light. Which is also part of the reason why many people have started to speculate that it is kind of the neural basis for path integration, because you obviously can perform path integration also in darkness, right? So if also as a human, like if you close your eyes, if you walk around, you always still maintain kind of a sense of where you are, and so it does work in humans as well as in rodents. But how naturalistic these environments are in rodents, I don't know.
Yeah, I think if I remember correctly, I think when I talked to Kate Jeffrey, I think she might have mentioned that something like grid cells. I hope I'm not talking nonsense here, but I think she might have said that they did a study, and kind of when you turn the lights off and rats and their square boxes, that the grid cell pattern just gets weaker or something over time, and then when you turn the light on again, it gets back to normal. I think, take this with a large grain of salt, but I think she might have said that on my episode with her. But yeah, I don't know. Sorry, did you want to add something to that, or?
No, I think it makes perfect sense. It's in line with what I think, what I would expect. So, I mean, definitely, we also know, also as humans, we know it's, harder to keep track of where you are when you close your eyes, but still you're able to do it. And, it does make sense that maybe that's too easy explanation, but it does make sense that, like you see a similar pattern in rodents. If you're assuming that grid cells are the source of path integration, neuronal basis for that, it makes sense that they do function also in darkness, but also maybe the reduced activity is kind of an expression of, more uncertainty, which in the end leads to maybe worse behavior. We don't know, but I think logically it makes sense.
Yeah. I mean, I think in this hypothetical or potential scenario that I mentioned with the rats in the dark and in the light, I think one reason I would be curious to see whether, or I'm assuming that this was probably in a fairly boring square box, but if they had like different olfactory cues around the place, then I could imagine that the effect would be less or something like that. But yeah, this is, again, I'm not sure whether she said this in the first place. This is the funny thing, going back to what we talked about in the beginning, like, there's just so much we don't know about this stuff.
Absolutely.
It feels the first time you hear about it, it's like, okay, we figured this out. Like, we know how spatial navigation works. We've got place cells, we've got grid cells, and then you actually try and you realize there's almost nothing that we know about it.
Yeah, I 100% agree. I mean, I feel like that not only about, grid cells or navigation. I feel honestly about the whole neuroscience field is like, and that's what makes it so exciting, because every new paper or every question that we're trying to address kind of opens up 12 more new questions, right? Which are maybe even more exciting. So it's a good thing, but also it shows us all the time that there is still a lot we don't know yet, and there is still not a lot of work necessary to really understand what's going on, and to then hopefully help when systems like don't work so well, like in old age, or when it comes to interventions or disorders. So yeah, a lot of work to do.
Yeah, last point on this, I remember when between my bachelor's and master's, I was doing like an internship. I remember talking to someone who did, I had a bachelor's in psychology, but not real neuroscience at that point. And I remember talking to someone who did some sort of neurocomputation at a cellular level, something like that. And, asked her what she was doing. She said, that kind of stuff. And I was like, oh, well, I thought you knew how that worked. I thought, if you've got your action potential, like, what do you need? She's like, well, it's a bit more complicated than that.
Yeah.
But yeah, I guess we should probably start moving on to the next stuff, otherwise we'll never get to it today. But you already mentioned one point that, actually, you said you listened to my interview with Jakob Bermond, right? So You probably heard your name then, right?
Yeah, no.
Now I'm going to ask that question. So basically, I asked Jakob whether he knew whether there's a difference between, basically, would we have more grid cell firing or stronger grid cell firing when people are actually moving, relative to when they're maybe, lying in a scanner and only seeing it on a PC screen or imagining it. And Jakob said, I don't know. Why do you ask? I mean, you're going to talk to Matthias Stangl soon. Maybe he knows. So, do you know?
The sad answer is I don't know.
Okay. Yeah.
But the silver lining is we are on a good path to hopefully find it out soon. Because what I've been doing during my postdoc is to work with a very unique group of patients where we kind of finally get access to these signals, right? So to get access to deep brain oscillations, deep brain recordings while people are moving. And so that allows us to address these questions, but still we are like at a very basic level. So we do develop paradigms and techniques and experiments where we test these kind of things, but still this is such a new area that we're just at the beginning trying to basically make this work, like also from a technical perspective. And we now are at a stage where we can really start answering research questions, but we're at a very early stage. So we do see these grid-like patterns, for example, or other things that we'll probably talk about in a minute. We do see spatial representations, but we have not and haven't yet directly compared this between like virtual reality and ambulatory movement. If I'd have to speculate, I do think, based on the data that I've seen so far, I do think that signals are stronger when participants are moving. So for example, I have analyzed the same signals in terms of grid cell-like representations in fMRI, what we talked about before, and now looking at brain oscillations with electrophysiology while participants are moving. And the signals are very, very strong that we see while participants are walking around. But at the same time, there are so many other variables that could cause this difference.
I mean, you have a different imaging modality to begin
exactly
with. Right?
Yes, exactly. Like basically everything's different, right? Yeah. So it's in the one case we're measuring like hemodynamic response of the brain. In the other case, we're measuring brain oscillations. And yes, in the one case they are like lying still in a scanner and just looking at a screen. In the other case, they are walking around. But I'm not able to tell you if like the fact that we see stronger signals when participants are walking around, whether that's really caused by the fact that they are walking around or it's just electrophysiological methods or many, many other things that are different.
Mm-hmm. If you can't tell me this, feel free to just skip this question. But are you directly working right now on comparing physical movement to like computer-based movement or imagined movement? Or is that more... I wasn't quite sure like how to interpret what you said earlier, whether this is something you're actually working on or it's more kind of an intuition based on having worked on the different parts.
I'm not working on this particular question, so I can't really say much about it. But what I meant to say is that we finally have the technology. We, as in not myself, and maybe not our research group in general, but the whole field is now able to look at these signals and answer these questions. But it's not something that I'm explicitly working on.
I think we should probably start talking about the paper, otherwise we won't get around to it. So, I mean, I guess we've already mentioned quite a lot about kind of the circumstance in terms of you're working with patients here who are actually moving. But yeah, maybe as before, brief summary. What did you do? What did you find?
Sure. So the previous studies that I've done myself or in the whole field, tells us a lot about navigation in general, like how do we keep track of where we are, the neural mechanisms. But one thing that we also have to acknowledge is that a lot of these studies, almost all of these studies, come from, neural measurements of brain activation that was measured in not very naturalistic environments, not very naturalistic navigation scenarios, right? So most of the studies have been, including my own work, most of these studies have been done like using virtual reality while you're actually not really moving, which is, a question in itself, whether that's like navigation at all, right? So most of people are like in hospital beds while their brain activation is recorded and they do not move. They basically play a computer game on a computer screen, right? Or they are in fMRI scanners where you're kind of laying on a kind of a horizontal plane and look at a screen. So it's a question how this... I'm not saying so a lot of the findings that come from these studies are really, really important and very exciting. But still, we also have to see that we have to ask the question to what extent this translates to actual physical navigation, to walking around in the real world, to not being in a virtual environment, but being in a real room, maybe also with other people. That's another important aspect of real world navigation, right? You're not like playing a computer game where you drop off like a soccer ball in some corner of a virtual room. It's like you have many more variables. And one of this is, for example, the interpersonal aspect, that also hasn't been addressed a lot in previous studies, where, for example, if you walk along a hallway, you do not only have to have a representation of where you are, but you also need to know where other people are in order to, avoid running into them or these kind of things. So these are the two key things that we wanted to address here. First, investigate spatial representations and mechanisms of keeping track of where we are in the real world rather than, virtual reality and virtual environments, and then also see if the same mechanisms that we use to keep track of where we are, whether the same mechanisms also allow us to keep track of where others are in an environment. It is hard to study, or it has been hard to study for a very long time because of, as I said, technical limitations, right? So it's just really hard to get to these brain recordings when people are just walking around in the real world. But here, when I started doing my postdoc, I had this really amazing opportunity to work with a very unique group of patients. I said that before. These are patients who have chronic implant, and this is a device that is implanted in their head chronically. That means like for years or even decades. These are epilepsy patients who have a very severe form of epilepsy. And the idea is that this device comes with typically depth electrodes that are implanted in deep brain regions or in regions where their seizures might originate from, which very often is hippocampus, entorhinal cortex, medial temporal lobe, which are the regions also where we are interested in a lot, because that's where the navigation centers are kind of located also, or many of them. And so these epilepsy patients have these electrodes with the idea is that it helps them. So this device detects continuously, like 24/7, detects their oscillatory activation, their brain activation in these brain regions with these electrodes. And when it detects abnormal activity, then it's delivering stimulation in order to prevent seizures. And this helps a lot of patients that I've seen. It helps them a lot. Like, these patients are really able to go back to their jobs or have very, very normal daily lives. But so it's really good for them, but it's also like a very unique and great research opportunity because we finally have access to deep brain recordings of regions that are really essential for memory, for navigation, for all of that, while participants are, doing all kinds of normal daily life tasks. They can do everything. They can just walk around and have a very normal daily life. The problem, though, is, as I said, this is a clinical system. It has been implanted not for research, but for clinical purpose. So we don't have easy access. It's not like you can plug in, like a USB stick and get these data. It's you have to work really hard. And so one of the very first things when I joined this group of Nantia Sutana at UCLA, one of the first things I was involved in was to develop this technical platform, so that even allows us to get real-time recordings and also access to the stimulation capabilities of these devices, which then finally, in the end, really allows us to do these experiments. And that's what I said before. We can have access to these signals. We can record them. Now is really the time to think about, experiments, to ask questions. But there was a lot of technical development necessary to get to this stage. And there is also a paper in Neuron from Uroš Topalović from our group, which basically just talks about the technical development of how do we do that. And it's not trivial, but that was kind of the prerequisite for everything.
The first half was an engineering job, and then you could do science after that.
Exactly.
And just very briefly going back, that your background in programming probably didn't hurt either.
Exactly. It helps with that kind of stuff. And I mean, in our group at UCLA, Nantia Zutana, like she manages to put together like a really, really awesome team of people who are, like engineers and people who run experiments, who design experiments, but also there is a lot of technical development involved. So like programmers, electrical engineers, like literally building physical devices, it's not something that you do on a daily basis, like in human, fMRI studies or whatever. But there is a lot of technical development and engineering necessary to run these studies, and that's what this group has done. That's what I've started to do when I joined this group, and it finally allowed us to address research questions, which is that was a very long introduction to get to the actual.
I mean,
to the actual paper. No.
Yeah. So anyway, so you, so you've got now the patients. We know what they're doing. They can move around. So what was the finding?
What we finally were able to do is really look at these brain waves during real world navigation, during just walking around physically in a room, right? And what we wanted to do is we wanted to see how our own location, or how the human brain during real world navigation represents location. How do we know where we are? What are the neural representations and the mechanisms that allow us to keep track of where we are? And one thing that we wanted to look at in particular were like theta oscillations, because it has been shown multiple times in other studies that theta oscillations is just a very prominent oscillation, relevant for many things, including memory, including navigation processes. So we wanted to look at how these theta oscillations actually help us to keep track of where we are. And what we found in this study was that when participants were walking around and we measured their brain waves, so to say, their theta oscillations, while they were walking around, we found that the oscillatory power, like the amplitudes of these oscillations, they got stronger depending on where they were in the room. So it basically was a representation of their location. Specifically, theta oscillations had a stronger oscillatory power when participants were closer to a boundary, which in a way is a kind of a code for where we are, right? Or where participants were. But that was only the first part. And the second part of this study was we asked participants to not only walk around, but also at a different task. We asked the same participants to basically sit in the corner of the room and watch another person while this other person is walking around, which was me in this case, which was a very funny feeling, by the way, to just walk around in a room being watched by someone for like an hour. And I'm sure it was not a very exciting thing for participants.
I was about to say, it must have been not much less weird for them.
Yeah, no, absolutely. I'm not sure who felt more strange, but anyways, it was a funny scenario, but I mean, it worked out really well, because what we did find is that participants used the same code to represent not only their own, but really my location in this case. So they did not move at all. They were just in the corner of the room, but their brain waves, their theta oscillations were modulated but where this other person was in the room. And this was just a really exciting finding, because it was the very first time that somebody could show that, someone's brain activation is actually influenced by another person's location. So, and this was a very, very strong effect. We see that in every single participant. So it seems to be like a really strong, finding, that we have here.
I mean, is it fair to call it a boundary vector cell independent of person near the boundary, or is, I mean, I'm not entirely sure what the definition of boundary vector cell exactly is. But
I see. So I would not call it like that, but it doesn't mean it's wrong. It's one possibility. But at the same time, the signal that we measure here stems from, like many cells, like again, populations of cells.
Right. It's, to clarify, you're using intracranial EEG, not
Yes.
single
It's not
cell
single
depth
cells.
recordings or whatever. Yeah.
Exactly. Yeah, I should have mentioned this more clearly, but it's population. Its local field potential is probably from many cells together. So it's the sum signal of many cells, and it's possible that this signal comes from, like it has been shown there are what you said, boundary vector cells, or there are border cells. But there are also other possibilities. It's possible that it comes from place cells, which also it has been shown that place cells tend to cluster around, more boundaries or maybe like objects. And this is another possibility that the walls that we have used, or not that we have used, the walls of this room in this experiment, right? There were just things on the wall. There were visual signs on the walls. There were also things like, like a Wi-Fi router or something like that. It could be that it's actually these objects in the room that were driving this response. We're not sure whether it's exactly the boundary per se, or that it's something that is like on or near the boundary. So I wouldn't go as far as saying it's boundary vector cells. But it's obviously reasonable to assume that this contributes to the signal. But with this kind of population level signal, it's kind of hard to disentangle and really say what cell type is driving it. That's something for, then again, maybe going back to single neuron recordings in stationary participants. Like this is done, for example, also in epilepsy patients. While they are in the hospital bed, you can get access to single neurons, which would be a very exciting next step to do that. Or hope for another key technical development step, which would, hopefully at some point allow us to get not only intracranial EEG, but really single neuron recordings while participants are walking. And I know that there are people working on this, and I know that hopefully soon we will have access to this data, and then we can really, pin this population level finding down to specific cell types, also be more specific about brain regions, these kind of things.
Maybe I'm curious, was the initial plan of this study to start also with someone else moving in the room? Because it seems like, there's this huge kind of technical setup to get this to work. And then you kind of did two things, right? You did the one thing is the people moving through the room. And then in the same study, you also did watching someone else move through the room. Was that kind of planned from the beginning to not just do, in quotation marks, just to not just do one of the first, I don't know, is this the first study to have people moving freely and get recordings? Okay. Yeah.
No, it's not the very first. So there has been work, for example, from it was Sarah Agajan, also in this lab. So I would say it's fair to say that this lab, Matthias Stangl lab at UCLA, is one of the pioneering labs where this has been done. But the study that I was involved in here was not the first one. So it has been shown, for example, that movement speed is modulated, or that theta oscillations are modulated by movement speed when you are physically moving, and that has been shown
in humans.
In humans, exactly, with a very similar setup. The setup gets better and better though, so we are now able to really, even as a next step, even go outside, walk in, like in the real outside world, outside the lab, and so it's getting better and better. And also there were some improvements in my study compared to what has been done before, and there are also new improvements for future studies. But and actually to answer your question, so it has been done before. So this time it was not only the focus to have someone, walk around and measure the brain activation. It was really the key idea to compare spatial representations between self-navigation and when you keep track of another person, to basically answer the question of, do we use the same mechanism to keep track of where we are and where others are, or how are the parallels, how are the differences?
By the way, is the signal equally strong for self and other? Or like what's the relationship there?
Yeah, it's not equally strong. I mean, it's also, again, it's not a very fair comparison, because when you're walking around, there is a lot more involved, like, you have this motor activity and all of that. So it's not a completely fair comparison. But what we see is that the signal when you observe someone else is typically, there is more variability. Plus, one finding of this paper is also that you do not see exactly the same frequencies that play a role during self-navigation compared to observation, because that's shown in the paper is that when you walk around yourself and basically your brain activation is modulated by your own location in the room, we see that this effect is happening on a pretty broad frequency bandwidth, like around, say, 3 to even 12 hertz. So we do see the same effect also when you keep track of someone else, but we see that the frequency band where we see this effect is narrower. We see that here only in around like 5 to 8 hertz. So it could be now that the difference comes from the fact that in one case you are moving, in the other case you are sitting, but it could also be that it's, just a qualitative difference, or just because the signal is maybe weaker when you're keeping track of somebody else. It's something also that are good questions to address in future studies, and we are not exactly sure where that difference come from, but we do see this difference. So it's not an identical signal, and it's also not exactly the same strength.
Yeah. Okay. It seems to me as if you've kind of found something you really enjoy in a place that you like being in, but postdocs positions tend to not be unlimited. So kind of what's your plan there? Is do you have lots of time to do more studies, or is it a kind of thing where you have to like stop in the middle of it and let other people do that?
So first of all, your guess is very right. Like I really like where I am now. Like, this lab is really fantastic. It's great people, like Nadia, the PI, she's really great. And the opportunities we have here are unique, really awesome. So yeah, it's just a really, really good place to do science. And, I would like to stay. Also, I think we're in a good position in terms of funding, so that we can continue with this line of work. So it seems what we do is interesting enough for many other people and funding agencies that we hopefully get, and it looks good. So. And also for me personally, it looks like I want to, and I will hopefully be able to continue this line of work. And, this was really a first step, like first the technical development, and then looking into, this boundary related
my first step, I don't think is going to lead to a Nature publication. It's a good first step.
You never know. I wouldn't have expected.
I'm looking forward to your second step. that's good.
From here it can only go downhill, basically. So that's the other thing. No, but this was very exciting. But I mean, so many things came together. Like Nadia is like a really fantastic mentor. The opportunities we have here are just really great. Literally every single person in this lab is just really, really good and really, really nice. So
that's pretty
amazing. Just having these opportunities to work with these participants who are also really, really great. They really want to do this. They really want to help us doing this work. So it's just a very exciting place to be. But I also think this is the secret recipe for why these things really work out, right? So having such a team, having such a PI, having all of these resources really makes this possible. And then, of course, we were also not expecting this to be so strong and so exciting, but maybe a bit of luck involved as well.
Yeah. I mean, I guess I had like one question, which is related to some of the stuff we've mentioned. And I guess you've kind of indirectly alluded it to by mentioning that funding right now is working well and people seem to be liking the stuff. So, I mean, it's been now, almost a year since you've had a first author publication in Nature. So what's that like? Is it, I mean, for me, Nature is always the most prestigious journal. I think for me, a fairly clear number one. And so, I don't know, is it as good as you expected? Or, I don't know, yeah, what's it like?
That's a very good question. It's
I'm assuming I'll get there multiple times soon, but so what should I expect?
So it was an incredible learning experience for me. I have to say though that the experience publishing in this journal was not essentially different to other journals. It was like, the current biology paper was also very well received, and I really loved that paper. And so it's, I don't know what to say. It was just, I also felt it's more pressure to be very honest.
To have that, to be a person who's published in it, or to try and get it published?
Yeah, to try and get it published in such a journal, because, once first you submit it there and you just hope that people like it, but at some point you get reviews and then you realize, okay, well, there is an actual chance to squeeze it in there.
It's like, oh no, don't mess this up.
Exactly. Yeah. And I mean, I worked like this was actually during COVID time. So I worked in home office literally like 24/7 to work on these reviews and to work on everything the reviewers wanted to see. And, so it was a lot of pressure, but it was, at the end of the day, obviously it was a very exciting experience. Also very rewarding. There was a lot of attention also after we published that paper.
I mean, that's how I found out about your research, right? Like even the other stuff that we talked mostly about today. Yeah. I hadn't read that actually until then.
Yeah. No, I agree. But also I think, I'm not sure to what extent this is like about the journal. I think what we did here was just really, really exciting for many people, because what I said, it's basically the first time, or it's maybe not the first time, but it's one of the first times where we can have brain activation measured in participants while they are just walking around. And then we can have brain activation for, like when you are in a more like social or interpersonal context where you keep track of somebody else. It's just questions that are just really, many people are probably interested in, but based on so many limitations, you can't really address them very well with, or it wasn't really possible before. And so I think the novelty of this, and then obviously the findings were also really new, really exciting. So I think that caused a lot of attention just based on what we did, and not only where it was published, but yeah, obviously it's also for me as an early career, it's a good thing.
I just saw on some UCLA website there was just like a little profile about you or something like that, and I'm not entirely sure what it was, but there was one of the sentences I'm just going to read. It said, "He has established collaborations with the National Aeronautics and Space Administration, NASA, and the European Space Agency," what's it called? ESA? I don't know. "And serves as a co-investigator on prestigious projects to characterize neurocognitive consequences of space flight in astronauts and to identify neurobehavioral risks associated with future exploratory space missions and their mitigation." So, you have a meeting to attend, but can you say, like, in 1 minute, what's that? What's going on there?
Yeah. It's a little bit independent, but yes, also very exciting work. So that's mainly, I was asked by Alexander Stahn, who originally was at Charité in Berlin and now is at the University of Pennsylvania. So we want to look at how spatial representations change, for example, in extreme conditions, like, for example, when you are in isolation. And so we're working on that. So there is funding from ESA and NASA to look at, astronauts, they are typically isolated from the rest of the world when they are in space. And so this can be both simulated on Earth, like they are like for weeks in isolation. And we just wanted to see how that changes how that changes spatial representations and your whole, like, you know, perception of space and how you keep track of where you are. And we do the same thing also with astronauts at the International Space Station. So a lot of things change when you are in space. So when astronauts come back, a lot of things are not exactly the same as when they left. And I am particularly interested in looking into you know, their spatial representations and how this changes
from kind of having moved in 3D, kind of. Or,
exactly. Yeah, it's just a very
factor.
Yeah, we can also see that. So this has been done. This was not by me, but it has been shown already that, for example, being in space, you know, it has physiological consequences to your body. And one of this is, for example, that you see physiological changes in the hippocampus, for example, and so it makes sense to. And obviously, you know, your whole spatial perception and your whole experience being in space is different when you are in actual outer space. So it makes sense to look at how this impacts spatial representations in actual astronauts or just in these very extreme conditions.
Yeah. Okay, well, that sounds all very exciting. I think at some point I have to have you back to talk about your paper about the neural correlates of movement in space or something like that.